Enhancers are the key gene sequences for turning on and off the transcription of genes in multicellular organisms. Experiments with reporter genes in a variety of different genetic model systems have been invaluable to our understanding of enhancers. Multicellular organisms are made up of many different cell types which differ from one another because they use their identical genomes differently. Muscle and retinal cells make some of the same kinds of proteins and RNAs, and some different ones. One of the ways that these differences are achieved is through regulation of transcription. The cis-acting elements of genes that mediate transcriptional regulation are enhancers, and the trans-acting factors are transcription factors. Two types of transcription factors, activators and repressors, can bind to specific base pair sequences within enhancers. An enhancer is simply an array of binding sites for different activators and repressors. A gene's enhancer can be identified by preparing a series of genomic DNA fragments located upstream and downstream of the gene, and cloning them next to the promoter of a reporter gene construct. Because this gene is a mouse gene, we're using a reporter with a mouse promoter. Next, several lines of transgenic mice are made with each of the different reporter constructs. The lines transformed with a reported transgene that has an enhancer will express GFP. Scientists who first performed experiments like these found that the mice that expressed GFP did so in a pattern corresponding to that of the gene that the enhancer came from. For example, the enhancer of a gene transcribed in the adult eye will express GFP in the adult eye. The reason is that enhancers control the developmental time and cell type of gene transcription. Enhancers do so by integrating information from inside and outside of the cell through transcription factors. For example, suppose a gene is transcribed in eye and skin cells and has separate enhancers for each. In addition, expression in the eye occurs only in S phase of the cell cycle. How is this specificity achieved? First, in skin cells, the activators that bind the skin enhancer are all present and modified appropriately so that all of the activator binding sites in the enhancer are filled, and the enhancer can activate transcription. In eye cells during S phase, the eye enhancer's activator binding sites are filled by a different combination of transcription factors that are present in the eye and modified appropriately during S phase. Transcription does not occur in the eye during G1 phase of the cell cycle, for example, because one of the activators is not expressed. Another one of the activators is not phosphorylated, and so it cannot bind the enhancer, and two indirect repressors interfere with activator binding. Expression of the activators, the repressors, and the activity of the enzymes that modify each of these proteins can depend on the stage of the cell cycle, many other intracellular events, and also on signals from other cells or other environmental factors that affect signal transduction pathways inside the cell. Using reporter genes, scientists discovered other properties of enhancers as well. For example, enhancers can work when at either the 5' end or the 3' end of a reporter, regardless of whether their normal genomic location is 5' or 3' to the promoter. The orientation of the enhancer relative to the promoter makes no difference either. All of these properties of enhancers make a powerful gene identification technique called enhancer trapping possible. Enhancer trapping is a method for identifying genes on the basis of their expression patterns. Suppose you're interested in finding genes involved in mouse skin development or function. Some of these genes will be transcribed specifically in the skin. You could make lines of transgenic mice, each of which has an enhancerless GFP reporter gene integrated into some random location in its genome. Rare lines will express GFP specifically in their skin. In these lines, the reporter must have, by chance, inserted near an enhancer activated in skin cells, and that enhancer must normally regulate a gene transcribed in the skin. You can use the inverse PCR to identify genomic DNA sequences adjacent to the transgene insertion point, and compare that sequence to the mouse reference sequence to identify the gene.